Hello and welcome to Inside Exercise. I'm Glenn McConell. Today I've got Professor Paige Geiger from the University of Kansas Medical Center. We're discussing all things heat. So heat therapies, the effect of that on your insulin sensitivity, on other chronic diseases, comparing and contrasting the effects of heat, which raises your core body temperature, to exercise, which also raises your core temperature. So, how much of the effects of exercise are due to the heat. We talked a bit about mechanisms. So, for example, the heat shock proteins that appear to be important, how much heat you need, for how long and how often, and what's the washout rate. We discussed, is it possible we're just living in too much of a comfortable environment? It's always plus or minus 1 degree Celsius. Is that possibly detrimental to our health? People have been saying saunas are good for you for hundreds of years. I guess the science is only now catching up. It's a really interesting area, and it's clear there's lots more work to be done. I think you'll really enjoy this one. Hello, Paige. Welcome to Inside Exercise. How are you?
Hi, great. Good to see you.
Yeah, good. I was just thinking, I reckon Paige Geiger is a really cool name. Something about that.
Yeah, maybe. I don't know. Lots of vowels in that name.
Lots of vowels. I guess Paige is not really a name we hear down under. So yeah, so it's the morning here, 7 a.m. What's it for you over there?
About 4 o'clock.
4 o'clock. And you're in Kansas City, Missouri. Kansas City, Kansas.
Kansas City, Kansas. That's right.
Kansas. All right. Okay. So how did you actually get into this? I mean, some of the people I've had on, quite a lot of them actually, were formerly athletes, sports people, whatever. And then other ones obviously started off scientists and then went into exercise science. How did it work for you?
Yeah, so I definitely come from that exercise camp of being an athlete. I was a rower in college and always been a runner. Went into graduate school and studied muscle physiology. But when I was finishing my postdoc with John Halsey at WashU, I think I was, his 80-something postdoc. And so at the time, thinking about what could be an original line of research coming out of that group of fantastic scientists, it was tough to come up with something that hadn't been done and that was original and that was really exciting. And so I came across a review article that Phil Cooper from Colorado had written. And Phil was a physician out of Denver who he told me when I met him later in life that he was bored with his private practice at the time. And he just decided what would happen if you put a bunch of people with type 2 diabetes into a hot tub for several weeks. And so he did just that, wrote up a small paper on it, managed to get it published in the New England Journal. And he put these people in the hot tub for 3 weeks and didn't change their diets, didn't change their activity levels or any kind of medications. And at the end of the 3 weeks, they had improved blood glucose and lowered HbA1c levels. And so he, at the time, thought that was due to improved blood flow. But he went on to speculate that maybe the heat shock proteins, which were activated with the heat, had some role in preventing oxidative stress and inflammation that we know can interfere with insulin action. And so at the time, I was looking specifically at insulin signaling pathways in muscle and oxidative stress and things like that. And so I don't know, it just seemed like an avenue of research that I could pick up and get started and just seemed really interesting to me at the time.
That's an amazing story. And the thing, well, actually a couple of things. It reminds me of myself when I finished my PhD, so it's a bit earlier. I was with Mark Hargraves at Melbourne University, and he was the big name in carbohydrate metabolism. So I'm like, what am I going to do now? So I was literally looking through the Journal of Applied Physiology. So that is stressful. I'll go, I think you'll vouch for that. And you've obviously come up with a great thing there. But the Phil Hooper thing as well, that's an amazing story.
Yeah, it was really, and what's interesting about the field, Glenn, is that I think, that Phil's paper came out in 1999, where he did this small little study and put these people in the hot tub and I was starting my research lab in 2005, so not too long after that.
Yeah.
But the field, instead of going to everyone get in a hot tub, this is the cure-all, the field really went to what are heat shock proteins doing? What are the cellular processes? What are animal models that we can use to study this? And really, it wasn't until 2015 when Chris Minson from the University of Oregon published his first study looking at hot water immersion in young, healthy college students, probably from around campus, that people are now, everyone is turning to look at using this intervention in people. But really, there's been almost 2 decades worth of work done at the cellular and basic
That
science
is
level.
really interesting, because I actually first heard about this because being in Australia, Mark Fabreo had done his studies with wrapping up, rats and mice and in heat vests and whatever, and looking at the heat. I actually thought that was the first. And then I realized later that Phil Hooper had put his people with type 2 diabetes in there. And to be honest, I didn't know where you fitted. I was like, hang on, didn't Mark Fabreo already do that? But it was actually the other way around. Yeah. So that's interesting yeah. So if we just step back a bit, because I just think I met Phil Hooper at a conference, and at one stage we were talking about collaborating with nitric oxide and seeing if that was playing a role. I just think that's amazing, that because he said, as you mentioned, he was just bored. He's a GP and he funded his own studies. He said he never got a cent for this. He would just put his own money into it and do the studies,
Yeah.
which was just pretty amazing, right? And he's so enthusiastic and all that. So
yes, he is. And I'm really glad that I didn't know all that when I based my entire research program on those couple of papers.
yeah. Wow.
Because if you think about it, the papers that you write, sometimes you feel like these small studies go out into the void and you don't know who reads them or what the impact is, but it was the start of my entire, independent research career was based on some of his ideas. And then, there's been many more since then. So yeah, you just never know where your study's going to land.
It's worked out well, obviously. I'm just interested though, like, how did he even, I don't know, I need to get Phil Hooper on here. How did he even have the idea initially? Do you know? Like, is it because people near the equator have better insulin sensitivity? Like, what was the idea even that getting hot? Why do you even think to throw people in a hot tub is what I'm getting at?
Well, according to Phil, he was sitting in a hot tub literally when he got the idea.
Okay.
And I think Phil is one of these people that's sort of like a know-all Renaissance man type, so he started thinking about how heat has been used for centuries. It's really an ancient approach to health, thermal spas and things like that. So I think that's where he started and just thought, well, maybe improving blood flow with heat could clear glucose. I think it was just that simple.
Okay. All right. So we'll talk about that later, that it's probably more the heat shock proteins, as you touched on. But that's interesting. So just stepping back a bit, even, is there evidence? I don't know. I honestly don't know any of this. So if you're closer to the equator or like in summer versus winter, is there any evidence that insulin sensitivity is different? I don't know.
No, and this is an interesting aspect of this whole story, is that if it is truly heat shock proteins or some sort of heat induction response that is beneficial in terms of metabolism and other chronic diseases, then the basic or sort of constitutive level of these proteins may be higher in people that live in warmer climates, but you still have to activate these systems. So heat shock proteins have this whole feedback inhibition, like most systems in our cells and our bodies. So if they've been activated, then they get turned off. So someone living in a warm climate is going to be protected from constantly having these heat shock proteins activated. And so they adjust. And so there's really got to be a stimulus, much like an exercise stimulus, to get these heat shock proteins to have these beneficial effects.
Well, I guess if we just step back a bit. So we're talking about, I guess, the mechanisms there, but just purely like if you're near the equator or in summer, is there any evidence that you're more insulin sensitive, for example? Or is that not really something?
No.
Okay, all right. So, so let's talk about these heat shock proteins, because it'll be new to some people. So naturally you think of it and you go, okay, heat shock. So if I have a really high heat, it's a shock to my body and these proteins turn on or something like that. Is that, why don't you talk a little bit about these heat shock proteins, how, they've been around, obviously they've been around forever, but are they the same in different species and, things like that?
Yeah, so they're very highly conserved. And like most great things in science, they were found by accident. So someone accidentally turned the incubator up too high in a plate of cells and found these proteins that got, expressed. And so that's how they were discovered. And honestly, that's sort of why they're misnamed a little bit as heat shock proteins. It turns out they're actually activated by all kinds of stressors. They could be just called stress proteins. They're activated by shear stress, by changes in pH, by UV light, all kinds of things, and not just heat, but that's how they were first discovered. So they're in all systems of the body, they're in all different organisms, and they really are ubiquitous, and they're meant to be a cellular defense mechanism. So they help primarily folding proteins, misfolded proteins in stress conditions, but more and more research has shown they also have a lot of cell signaling mechanisms as well. And that's where our interest in metabolism is really started with what can they do in terms of interacting with proteins that we know are important for insulin signaling, and more recently, what do they do in mitochondrial function as well?
Right. Okay. And that was one of your early studies. Well, actually, I don't know if it was early or not, but one of your studies. So you put rats on a high-fat diet; they became insulin resistant. You, why don't you tell me what you did?
Yeah, and that's actually Mark Fabrio's lab did that as well. They did that before us, and yes, if you put mice or rats on a high-fat diet, give them sort of this diabetic state, 6 weeks or so, feeding them a 40 to 60% high-fat diet, they become insulin resistant. And what we initially did was, if we put the rats and mice in a water bath, so we have our little mini rodent hot tub in the lab where we put them waist down in the water, hot water. If we just do that once a week for 20 minutes, we can completely prevent the effects of that 6-week high-fat diet.
Wow.
Both in terms of glucose uptake in the muscle, mitochondrial function, and different insulin-sensitive tissues like the fat and the liver as well.
Wow. So that's just one 20-minute bout. Okay.
Yeah, I always tell my students it'd be similar, like if you ate at McDonald's a high, Western diet, high fat every day fast food for, every meal of the day, and then just sat in the hot tub 20 minutes once a week. That's basically what we did with these rats. So it's pretty potent effect.
So I guess, okay, sometimes with rodent studies and things, they make it a bit extreme, like I always remember the studies where they looked at exercise and they did swimming, and they did 5 hours of swimming with a weight on their tail. And you say, well, that's not particularly, likely that people are going to do. Obviously we don't have the tail, but so how hot did you make them? Is it something that we would, a human would never tolerate? Or?
Not really. So the rodent studies, we actually increased the body temperature quite up to 41 degrees, and going from 37 to 41. The critical temperature that we've been using in humans and that other labs have been using is raising the core body temperature 1 degrees Celsius. So that's really
okay. Because 37 to 41 in a human, that would be very uncomfortable. But 1 degree is not that bad. Well, how bad is that?
It's uncomfortable. It's uncomfortable. If I put you in our hot tub, Glenn, and made you sit there for the 30 to 40 minutes, which is about what we do now, you would be uncomfortable, but most people adjust very quickly. After 2 to 3 visits, people are very comfortable with it, and they really don't mind it very much.
Well, that's the thing I was thinking, because at first you think 1 degree isn't that much, but we're so good at thermoregulating, like it actually takes quite a lot to get up by 1 degree, because you get all your peripheral effects and everything. To actually get the core up is uncomfortable. And I think we all know, maybe I'm super susceptible to it, but if I have a bath, if I have it even slightly hot, it's not fun.
Yeah, so, you get uncomfortable. That's what I tell everyone is, when everyone's been in the hot tub for a little too long and you start to get uncomfortable. In our studies, you have to stay past that point. And your body does start to adjust, but our immediate response is, oh, this is stressful, or this is uncomfortable. I need to remove myself from this setting, right? And I think for some, this is completely anecdotal, but I think athletes are used to pushing past that point of discomfort, in a way. And it's similar to that feeling of being in the heat. You get uncomfortable, but you can do it.
Well, I was actually just thinking that's a beautiful thing, because obviously we're all into exercise. I remember the studies we did where we had fluid ingestion, no fluid ingestion or whatever. It actually didn't matter the first 30 minutes, whether you drank fluid or not, the body temperature always went up by 1 degree. It was always about 1 degree it went up. And then if you drank, it would tend to plateau out. And then if you didn't, whatever. So that's interesting. So with exercise, you're not even that, okay, you might be uncomfortable in other ways, but you could have a trained person, they're cruising for 30 minutes and their temperature's up on 1 degree. So I guess the question there is, how much of the responses during exercise are heat shock protein related and how much are unrelated and all that sort of stuff?
Yeah, I love that question because I think everything is heat shock proteins, right? So
Uh-huh.
obviously they're not, but I do think that maybe many of the adaptations that or the benefits of exercise could come from heat proteins. So, we've shown that they can improve mitochondrial function we've shown that they can decrease pathways of inflammation and things like that. And there's actually a lot of people studying sort of heat shock proteins and post-exercise recovery. So whether or not a pre-exercise heat bout, because you would induce those heat shock proteins, could then be protective against, say, eccentric exercise damage. Like those studies have been done. So, I'm sorry?
Those studies have been done or haven't been done?
They have been, yes.
Okay. Well, maybe we'll get to, actually, I want to ask you about that now. So what did they find? So
yeah, so if you can decrease some of the markers of muscle damage, creatine kinase, things like that, by that pre-induction with the heat shock proteins. But again, some of those were animal studies, and whether or not those translate to performance and things like that, I think there's still a lot to be done in that respect.
Wow. Okay, because that actually reminds me of preconditioning. They have a thing where
Right.
why don't you just say that for a minute, because I don't think I quite understand it, but wonder if that could be heat shock proteins.
Yeah, so you mean just like the idea of doing an exercise bout and turning on all those pathways
With the heart, they talk about preconditioning with the heart. Is that?
Yeah.
Yeah,
there's actually a lot of heat shock protein studies were originally done looking at cardiac studies, and so looking at ischemia reperfusion and things like that. And so having an ischemia response, turning on all those heat shock proteins and then letting them go and refold proteins and turn off inflammatory pathways was then protective. So yeah, I do think there's multiple things going on. There's sort of that preconditioning or protective effect before a bout, but also benefits to the actual bout itself, whether we're talking heat or exercise.
During the bout. Wow. Okay. So maybe heat shock proteins is regulating everything. No, I mean, I think we know that there's really complicated, physiology going on with exercise, and we also know there's redundancy. So in a couple of the earlier podcasts, we talked about, for example, during respiration, you can remove some pathways and your respiration's still normal. So I assume there's redundancy, but is there? So with exercise, for example, if you exercise and you manage to keep the temperature down, so just say you've got people exercising and they're in a cold tub or something, and you keep the temperature down, do they get less adaptations, less responses to the exercise?
I don't know what some of the specific outcomes, I'm trying to think in terms of, say, muscle damage or something like that. But I know that those studies have been done where they either, in a thermoneutral condition or something where temperature is controlled and you exercise, you still get the heat shock response. So because it's again, there's that misnomer. They're turned on by other things besides just the heat.
Well, that's a beautiful thing. That fits perfectly in with the redundancy thing. So the heat shock proteins are important. And as you said, in humans, if your core temperature goes up by 1 degree, they turn on. But if you prevent that, is that what you're saying? Then it'll turn on due to the other factors. So like maybe the shear stress, which is
exactly
for the audience, which is, the blood flow pulsating through the vessels. I don't know, maybe the change in pressure during the contraction. I mean,
yeah, and mechanical stress to contraction, changes in pH, and all the types of things that we know that occur with exercise in addition to that generation of heat.
Wow. Okay. So pH is the acidity. Yeah. So okay. So what about when you have all these things adding on? I guess so. If you have the pressure, the shear stress, the acidity, the temperature, do you get a greater activation of the heat shock proteins?
I've always wanted to do those studies, and it's like I've never quite had enough hands to do them. I'd love to do sort of an isolated in vitro muscle prep that you can control the temperature and use contraction plus and without heat and really get at sort of the molecular aspects of that. I think you would see an additive effect, but I don't know that those studies have been carefully done to this point.
Well, I know there's a guy, Aaron Peterson, because Victoria University I used to be at, but I'm still there. I'm emeritus professor. He was doing studies there. He was doing like people were doing weights in like normal temperature and then doing weights in the heat chamber. So I guess that's the kind of additive, I think, idea. So is there like an additive? So if you have the temperature plus the exercise versus temperature alone plus versus exercise alone, is there like an additive effect? I guess heat shock protein activation, but also on the adaptations maybe to the training or the improvements in the insulin sensitivity, for example.
Yeah, I think the answer to that is yes. I think timing could be important though. Again, getting at that idea of if you did the heat bout before exercise, for example, maybe you could get that additive effect. You know, you may hit a ceiling if you're doing them at the same time in terms of just how much you can induce these different systems. I think a lot of that we just haven't sussed out yet.
Okay. One thing I can't help thinking about with heat is, you know, how much is going on, you know, which we've already touched on. So I remember one of the first things I learned as an undergrad, which was this Q10 effect. So, you know, with a 10 degree increase in temperature, you get a doubling of enzyme activity. And you say, well, that's actually not very relevant to exercise, for example. So with a 1 degree increase, you get a 10% increase in activity. But I wonder how much of that stuff is going on is maybe heat shock proteins, you know, it's affecting other things.
Yeah, and you're getting into the weeds a little bit on some, you know, some of these almost biochemical pathways that I don't want to insult someone who's already done that work and I'm not aware of it, because there's a lot of work at the real basic biochemistry level on these heat shock proteins. They even, you know, they call them something different, and there's a lot of knowledge about the way they fold and the way the temperatures impact their function and enzyme activity and all of that. So I think that you're probably onto something there in terms of how that could translate to exercise. But I think that the problem in the field is that the people that know the nitty gritty of that biochemistry of heat shock are not at all interested in the translation to exercise or health. And so it's this huge void of people that can't talk to each other really. So
okay, well, that's why we have collaborations, you know, bringing people together. All right, so I keep thinking about this, the heat versus the exercise thing. Yeah, so is it the same? Because again, so much more is going on with exercise, right? You've got the heat, you've got all sorts of others. There's cardiac output and, you know, metabolism. So for example, the muscle, if you heat the muscle or if you heat your body, the muscle's not doing anything and it's not turning over. So I just had John Hawley on. He was saying, you know, the difference with safe diet versus exercise. With diet, you know, you're not turning over the substrates. So I guess it's the same with exercise. You are. I guess it's the same with heat. You know, there's different stuff going on with heat. You're not turning over anything in the muscle, but during exercise, you're turning over stuff. Plus, you've got the heat. So I guess you start wondering, you know, are the responses similar? Are the same sort of heat shock proteins activated, whatever? So maybe just a bit of background there. Like when we say heat shock proteins, it's plural, right? There's more than one heat shock protein. So without going too deep into it, are the same heat shock proteins activated with exercise and heat? Do they overlap? Are they additive? Are they synergistic? You know, whatever.
That's a great question. And I don't know that exhaustive studies have been done. Well, in fact, I know exhaustive studies have not been done comparing heat and exercise. There are a family of heat shock proteins that, you know, run the gamut of size and molecular weight. We're primarily interested in the ones that are about 70 kilodaltons or 60 kilodaltons, heat shock proteins 70 and 60. They are highly inducible in the tissues that we're most interested with exercise: muscle, liver, fat, the brain, things like that. I do know that they are induced and expressed to varying degrees in all these different tissues. So I do think that it's reasonable to expect that they would be induced differently with heat versus exercise, or in, you know, from tissue to tissue. So I think there's a lot of nuance to how they respond to exercise. We do know quite a bit about, for example, just in skeletal muscle, they're more highly expressed in slow twitch fibers. So muscle fibers that are more oxidative and have more mitochondria have more heat shock proteins, but the fast twitch fibers have a higher level of induction of the heat shock proteins. So we've done some of those studies in my lab. And if we look at the comparison, the constitutive levels are higher in slow twitch fibers, but the induction is higher in the fast twitch. And so that speaks to, you know, what are they needed for? Maybe they need the fast twitch fibers need more heat shock proteins in response to exercise in terms of an activation mechanism versus what they're doing at a basal level. So
maybe just to clarify that for some people, because, you know, constitutive and things like that, some of those words. So you're basically saying the slow twitch muscle fibers, which are the ones that are more important for endurance, they've already got like a bit of a high basal heat shock protein level. And then maybe they don't get activated as much, so you don't get as much of an increase with the stimulus. But the fast twitch are lower, and then they've got, you know, more room to improve, I guess, so they get activated more. Is that right?
Yes.
Yeah. Okay. And again, just I guess getting back to, I was thinking about the dose. So I'm kind of caught up on that. So you're saying 1 20-minute bout, but that's uncomfortable, yeah? So, you know, for humans, the mice, whatever, it's like, sorry, but you just got to put up with it. So I guess what would you suggest therefore, like, you know, one uncomfortable sit in the hot tub, or, you know, a few, you know, sort of comfortable? I guess if they're not comfortable, they won't work. So how would you suggest practically people doing this?
Yeah, well, so the 20 minutes is what we've done in the rodent studies. The human studies, really, we've kind of based our work off of those original pilots that Chris Minson and his group at Oregon did. And initially, they actually used a really heavy hammer to make sure they saw something. They did like 90-minute bouts of heat. And when I say heat for humans in a hot tub, we're talking 104 Fahrenheit, which is about as high as a commercial hot tub will go. And they were putting subjects in for 90 minutes in the 104 degree, which I have done. We did one study where for 90 minutes, it's really long, but it is doable.
Your skin gets all wrinkled. Your skin gets all wrinkled up.
Definitely. Yeah. They have since backed off of that, and his most recent studies have used more like a 40 to 45-minute protocol. And that's now what we are using in some pilots at my institution. And so the general gamut that we've been doing is getting in the hot tub, and you get in and the water level is up to your shoulders. So it allows you to warm up quicker, and that takes about 15 to 20 minutes, and that raises the core temperature that 1 degree. And then you stay there for about 20 minutes at that elevated 1 degree core temperature, but we can bring the water level down, and it's much more comfortable. We set people up higher, we put a fan on them, and I stay like that for another 20 some minutes for a total of 45 minutes. And so you're at that 1 degree about 20, 25 minutes, total of 45-minute bout. That's where we're at right now. And we're doing those about 3 times a week and trying to get to maybe Chris's group showed that, you know, 10 to 12 sessions is what's critical to sort of start seeing some of these effects on the peripheral, you know, blood pressure and things like that. I'd love to see us kind of work backwards and figure out what the minimum level of heat is to see some of these beneficial effects, but we're just not there yet. Right now, we're still trying to figure out what we know works and then applying it to different patient populations and different disease conditions.
I was because I remember reading Phil Hooper's New England Journal of Medicine paper, which is, you know, again, is just a classic. So get people with type 2 diabetes, put them in the hot tub, and then as you said earlier, I think they had a lower HbA1c, which is the indicator that you know, that's sort of the last 3 months of glucose, the effects on the hemoglobin. You know, lower fasting glucose, I think. But didn't he only do like sort of 30 minutes? Didn't he just do it comfortable, like just jumping in the hot tub?
Did, but I think they did them every day also. So I think he did 5 days a week over 3 weeks, and we're doing every other day. And again, I think where the field is at right now is what is a beneficial dose and what is a sustainable dose to get people to come in for these studies. Like, it's pretty hard to get people to come in for an everyday study, as anyone that does exercise studies knows. So right now we're working with an every other, and it seems very tolerable, and it also is showing the beneficial effects on cardiometabolic health that we're hoping to see.
Okay, all right. And, you know, you've been an exercise person as well. I can't help thinking, if I wanted to activate my heat shock proteins, why would I get in a hot tub and get all hot and then turn on a fan and all this sort of stuff? Why not just exercise? You know, because you're getting the benefits of the exercise. And
yeah, absolutely.
is that the way you're sort of thinking as well? Like, you know, hey, you guys, you can do it this way, but if you want, not for your studies, obviously, because you've got to control it. But, you know, if you're just talking to your mum or your kids or whatever, would you say jump in the hot tub, or would you say go and exercise?
Yeah, that's a great point. And, you know, this is an exercise podcast, and I'm a diehard believer in the benefits of exercise. And I will always say, you know, exercise first. But, you know, the facts are that there are a lot of people that cannot do enough exercise to get the benefits. So whether it's age or another chronic disease, you know, you have to really get your heart rate up to see some of the cardiovascular benefits. You have to be able to do this consistently. So I think that what I'm really excited about is that heat therapy could be the alternative for people that can't get the benefits of exercise.
Fantastic answer. I was thinking that's perfect, because I'm thinking of able-bodied, you know, young or whatever. Well, you don't have to be young to exercise, but not injured or whatever. And I also thought that's a beautiful answer, and I'm sure you've said that in grant applications as well.
And in our pilot studies where we're trying to recruit people, you know, we're doing a study right now where we're recruiting individuals that are 65 and older that have some metabolic risk or metabolic history that puts them at higher risk for Alzheimer's disease. And, you know, that's a disease that is close to my heart. My mom has Alzheimer's disease. And I mean, it's a whole nother concept to think how difficult it is to get people with dementia to get the exercise that we know is beneficial for them. They don't understand why they should get on a treadmill. They don't remember from day to day that it's good for them. You know, so there's a lot of things that can keep people from getting the exercise that they need. And so, you know, sitting in a relaxing hot bath is something that is a little bit more palatable maybe for a lot of different populations.
No, that's a great point. Sorry to hear about your mum, by the way.
No, that's okay. She's still very active and healthy.
Oh, good. Okay, but that's a good way to bring up these other, because I was thinking, I keep talking about insulin sensitivity. That's sort of my background. So you just touched on some other things. So what are the other sort of chronic diseases and things that heat shock proteins do benefit?
Well, I think there's been not a lot of studies yet, but they're starting to increase. So there's been some work looking at anything to do with, you know, blood flow, peripheral artery disease, things like that. We know that there's very dramatic effects on blood pressure. So again, the Minson group has shown that 8 weeks of heat treatment is just as effective at lowering blood pressure as 8 weeks of aerobic exercise, at least in healthy young subjects. So there's a lot of potential there. We've done some pilot studies looking at the effects of heat on peripheral neuropathy, whether it's diabetes, or we've done a study in patients with fibromyalgia, which is a population of people that are very young and have very limited options for any kind of therapeutic measures to help with the pain that they experience. So we've put those individuals in hot tubs, and it's been dramatic how much better they feel after several weeks of the heat. They're more active, they sleep better, all kinds of things. So, you know, we're just kind of scratching the surface. And like I said, our interest in dementia and Alzheimer's disease has a metabolic slant to it. We think that lowering blood glucose and lowering blood pressure in those individuals could be preventative for Alzheimer's disease, because type 2 diabetes is a major risk factor for Alzheimer's. So it's still, I think most of the studies that are going now are still in the blood flow, cardiovascular, metabolic health realm. But some of the studies some of the retrospective studies, say looking at cultures like Nordic cultures, Finland, for example, has done some retrospective studies showing that people that use sauna regularly, which is, you know, a staple of their culture, have lower risks of dementia. And it's a sliding scale. The people that go 4 to 5 times per week, as opposed to 1 or 2 times per week, have much lower levels of dementia in the population. So I think there's, you know, the sky's the limit. We just need more studies. So I'll be new investigators and graduate students out there looking for something to study. The field of heat therapy is in its infancy, so we need lots of people to take on all these projects.
Yeah, they should contact you, huh? So yeah. That's the really interesting thing as well about the sauna, because that's obviously uncomfortable. I hate saunas. I know some people like them, but it's uncomfortable. So people obviously do things to do with getting hot that are uncomfortable. I mean, this sauna thing though is something that's been around forever. So are people using those studies much? So, when you think, how am I going to look at this? How do you choose between, the hot tub and a sauna? And I guess the hot tub's also going to have effects on your fluid balance and things like that as well. So.
Yeah, and I think it's a matter of where the field started versus where the field's going. So the studies were initially the hot water immersion. Now more and more groups are looking at sauna and infrared sauna. The time for sauna to get that increase in core temperature, it takes a little longer because you don't have that direct convective effect of the water, but you're going to get some of the same benefits certainly with sauna, and some of those studies are starting to come out. And what's exciting is, something that's been around for a long time in the athletic world is the use of diathermy to directly heat the muscles. So Rob Hidell's group out of BYU has done some really cool studies where they're just heating the muscle, and they can show a prevention of muscle atrophy with, sort of an unweighting model of inactivity. Okay. And so with just 8 days of diathermy, I think that's how long they did it for, they can inhibit muscle atrophy and also protect skeletal muscle mitochondria. So some pretty cool stuff with just direct heating.
I keep realizing you say cool and I say cool. We're talking about heat, so we've sort of got it back to front, but they are cool studies. So that's interesting, because I was thinking about that earlier. How much is the core temperature increased and how much is the temperature in the muscle per se? So you're saying they just heat the muscle and they get the effect. So it's not necessarily a systemic thing.
Yeah, and that's what's exciting about the field right now is more and more people are doing that. And Stephen Marrow, who's down at University of North Texas, is doing a really cool study where he's heating just the lower legs, so knee down in hot water, almost like hot water buckets, and older populations, and bringing down blood pressure just by doing that. So I think there's all kinds of range for utilizing heat depending on what outcome you're looking for.
Oh, so that's really weird, because I was talking about, so when you have the core temperature, you're heating up the whole body. So I was thinking if you heat just the muscle or, the leg, you would have effects just on that leg, which is interesting. But you're saying if you heat just the muscle or the leg without affecting the core temperature, you're still affecting your blood pressure.
Yeah, which gets back to your point earlier about, the differences with exercise and heat. And, with exercise, you are having this activation in the muscle where you're, using ATP and things like that. You've got this supply demand thing going that, it puzzles me what's going on with the heat. But it suggests that maybe the activation of the blood flow, what's happening in the smooth muscle, all of that is having some other, systemic effect. If you think about the dilation of the muscles in the lower limbs, I mean, those are pretty big vascular beds. And so that may be enough to see an effect in the whole body.
Especially with blood pressure. I guess the other thing is myokines, so again, people at home, myokines are proteins released from the muscle, basically. So we know with exercise, you get all sorts of myokines released that affect other tissues. And we know now that the liver's releasing things, adaptations and all sorts of stuff. All these different organs are releasing things. I guess if you're heating the muscle, then there's the blood flow effects, but also I wonder if the muscle's releasing any sort of myokines. Are people looking at that?
Yeah, absolutely. And this is an area that we're looking at actually. And Mark Vibraio's group, again, who's always on the cutting edge of all these things, Mark's looking at this. So there's actually a lot of work being done now where people are looking at the release of exosomes or extracellular vesicles from the muscle. So these myokines that you're talking about, they can travel in these vesicles, which are produced in all tissues of the body. But when they're produced from the muscle, I call them like a little bubble or something that comes out of the muscle. It contains everything that is inside that muscle. And guess what they contain? Heat shock proteins. So Yep. So we are actually studying that. What is in those vesicles? We know there's heat shock proteins in there. Where are they going with heat and with exercise? And can they have beneficial effects in other parts of the body? And that's, a growing area of research as well.
Okay. So if we just go back to the muscle, right? So we're talking about, well, I always think about muscle, but exercise, I was going to say exercise is muscle, but that's totally wrong. It's heart, lungs, every tissue. But if we just think about muscle for a minute, I just want to think again about mechanisms. So when the muscle's contracting, so you've got, if we just focus on the temperature changes, what's actually turning on that heat? Well, actually, I shouldn't just say temperature changes, because you said earlier, it's not just the temperature. What's actually turning on the heat shock proteins? Do you know? And what is the signal like right down there, at the protein level? You know what I mean? Is it, because I interacted with Philip Hooper at one stage, because he was hypothesizing nitric oxide, which is a gas which is produced during contraction in muscle, which I've done studies on. He was thinking that might be activating heat shock proteins. Do we know what's sort of happening down at that molecular level?
Well, I know that what turns on the expression of these heat shock proteins is the activation of heat shock factor 1, which is the major transcription factor. And it's kept in an inactive state when the proteins are all sort of just dormant and not active. And then it's turned on, and then it goes to the nuclei, where it helps transcribe it turns on, signals to make new heat shock proteins. And so when those heat shock proteins then are high, they actually then inhibit the activation of that transcription factor. Now, what I don't know as well is what comes before that HSF1 activation. What level of, are there heat, nerve receptors, things like that. I imagine there's a step in there that is involved as well. And I'm not sure that much has been done to look at that, again, comparing, say, heat versus exercise and so on.
Yeah, so that's where we was talking about nitric oxide, and we actually did a little bit. We haven't published it yet, but we gave like nitric oxide donors and nitric oxide synthase inhibitors and things like that, and looked at heat shock protein. So we'll just have to wait to see if that ever comes out. But yeah, so the heat shock factor 1, I know that gets like phosphorylated. So again, for the people in the audience, so basically you've got proteins, but the proteins themselves can be activated or inhibited by other things interacting with them. So one is phosphorylation, when you add a phosphate. Yeah. So do we know what sort of phosphorylates heat shock protein? What causes the phosphorylation of heat shock? Do we know what causes the phosphorylation of heat shock factor 1? And like the time course. So is it during exercise, for example? So rather than thinking about after exercise and turning on, the nucleus and messenger RNA and whatever, during the actual exercise, is there stuff going on? Is heat shock factor 1 being phosphorylated? Is it being translocated in the nucleus or whatever? Maybe people don't know this. I don't know.
Sorry. I'm trying to think in terms of exercise, what we know about that. We know that there are kinases, like, for example, GSK3, IKK beta, some kinases that keep that heat shock factor 1 in its unphosphorylated state. And when they're removed, it gets phosphorylated. So you could imagine that those kinases would be activated, say, with heat or exercise to remove that inhibition and then turn on the HSF1.
Yeah, because I guess I'm wondering, because we're talking about expression. So, you have the heat, or they have exercise, and then, hours later, there's an increase in heat shock protein 72 or whatever. I guess I'm wondering what's happening during the actual exercise and the heat, but I guess there's more to be done. Maybe that they get phosphorylated and then it translocates the nucleus and this turns on this and that. It's the sort of stuff that we were doing, and I know you were doing. I think I looked at your highest cited paper ever, I think, was PGC1 alpha. So, you mentioned mitochondrial biogenesis. So PGC1 alpha is a big regulator of that. And it's the same sort of thing, I guess, yeah?
Yeah, definitely. So there's it's what a lot of times I spin my wheels, which direction to go. I'm interested in understanding all those pathways, And I'm also really interested in seeing what the translation of these studies are. So, you know, if you only had all the time in the day to go both directions, but.
Yeah, because I know, because when I looked you up, I saw that you PGC1 alpha, which is a master regulated mitochondrial biogenesis, which is, you know, the idea, is increased with exercise, and this is a mitochondrial thing. So you've touched on quite a few times how heat shock proteins affect the mitochondria. So what goes on there? Do they increase this PGC1 alpha, or?
So we just did a study published a year or so ago with my last graduate student, Alex von Schulze, and that paper is published in Function, and it actually was in the liver with heat. But it was one of these, you know, I'm really proud of this project. It was very mechanistic, looking at the effects of heat in the liver. And, you know, it was one of these studies that you always want to do, and then you think, well, either I can't get this funded or I can't get it published, but we managed to do it. So we did a time course of what happens with acute heat, everything from immediately after heat up to 2 hours. And then we also did a chronic repeated bouts of heat over time in these mouse models to really see what's happening to the mitochondria as a result of this heat. So getting at this idea of the stress response and what it was doing. And it was really interesting. And I compare this to what we know about exercise, one bout of exercise versus repeated, you know, exercise training. And that information just wasn't out there on what for heat. And so in terms of the mitochondria, what we found was the acute response really is a stress response. It was decreasing mitochondrial respiration, decreasing all of the mitochondrial enzyme activity, and degrading mitochondria on a very acute one heat response. But then over time, repeated heat bouts resulted in improved mitochondrial respiration, improved mitochondrial efficiency. And the mechanism that was going on there was that of mitophagy, so the degradation of mitochondria. So we were turning on mitophagy with one bout, and that was getting rid of any kind of damaged or less efficient mitochondria, such that over time, when we gave the heated bouts every 72 hours over 3 weeks, then all that was left was the healthy mitochondria, and they became much more efficient. And we did everything from, you know, some RNA-seq analysis to look at all these different pathways that were being turned on in these two different acute versus chronic settings. And so we now understand a lot more about the way that heat induces a stress response, much like exercise does, but that over time, that's a beneficial stress response. So I thought that it turned out really great, and we're very proud of that work.
Wow, that's really interesting. So initially, so just to unpack that a bit, so you're saying with one exposure to heat or some stimulus that turns on heat shock proteins, it actually downregulates the mitochondrial function and enzyme levels. And then with repeated, then you get the upregulation, which is what you want, the improvements in function or whatever. Now that's a weird one, because, you know, we know one bout of exercise increases, for example, insulin sensitivity. And this has been this debate that, you know, like, oh, mitochondrial function and insulin sensitivity sort of go hand in hand. But yeah, this is actually quite an interesting one where it might dissociate. So with the one bout of heat, it downregulates the mitochondria. Does it also downregulate insulin sensitivity, or does it increase it? Because this is actually a nice way to tease that apart.
Yeah, that's a good question. So we didn't look at we've looked at markers of insulin in the liver, but we've done those studies in muscle, and one heat bout in muscle does activate and increase glucose uptake, much like one bout of exercise. So again, this may be tissue specific differences. This could be differences in actual, you know, glucose pathways versus mitochondrial pathways. I think, you know, even though exercise improves glucose, insulin stimulated glucose uptake, you're also getting all kinds of muscle protein damage with one bout of exercise, right? And you're turning on, you know, different you know, you've got reactive oxygen species being generated, things that you could call a stress response. In the same time, you're getting a beneficial glucose response. So I think there's just lots of things going on, and it depends what you're looking for. But
and there's lots more to study, yeah, because I did hear you say liver, but I didn't quite get it in my head. So yeah, this thing about the one lot of heat downregulating the mitochondria, that's the liver. So we don't know about the muscle, I guess. Is that right?
Right.
Yeah. I guess another thing is, what about washout period? So, you know, if you've had the heat exposure and you've had these great adaptations. How often do you have to do it? How long does it take to sort of wear off?
Yeah, that's a great question. So we did some initial studies in the rodent where we would activate heat shock proteins with that whole body immersion, and we would look at how long the protein expression took to go back down. Again, getting to this idea of there's an inhibitory response that these heat shock proteins get turned off when they're high, the activation is turned down. So we found that the protein expression levels stayed up for, say, 3 to 4 days, and then you could reactivate. And so initially, that's why we staged our heat treatments about a week apart, so that we were getting a full, we would get that response to be completely down, and then we would activate again. More recently, we did a study where we heated every 72 hours, again, in the rodent. The human studies, those really, I don't know that those have been done. So looking at heat shock proteins, for example, in the muscle, which is where we're most interested in them, would require taking multiple muscle biopsies over a time course, which no one wants to do. So I don't know that those studies have been done. We have looked at activation of heat shock proteins in muscle in humans 24 hours after heat, and we don't see them still up at that point. So we're not sure if we missed the window, we were too early or too late. But those type of studies about how long the protein is up or how long the heat effects last really probably are going to have to be looked at on a tissue by tissue basis and a population basis to really understand.
Okay. Interestingly, you saying population basis, it makes me think of, I had Claude Bouchard on a couple of weeks ago. So, he was talking about all sorts of things, but responders and non-responders and things. Does everyone respond to heat?
That's a great question. I don't think we know. Everyone has these heat shock proteins. Everyone has probably various levels of them. I would imagine that they vary based on things like, muscle fiber type. We know they vary with age. We know they can vary with chronic disease. So, there have been some studies, at least in animal models, looking at heat shock proteins with diabetes, and we know the expression levels are down, but they are still inducible. So I would imagine that even in people with different disease states, that they can still be induced, activated. But whether or not we see sort of a non-responder type population, similar to what we know you can see with exercise, I imagine we will. Maybe not non-responders, but not as, the effects are not as much, not as effective.
Okay. Now, one thing I was thinking as well when you were saying that was, you said earlier on something about you can eat sort of a bad diet or whatever, and then just have 20 minutes of, exposure to a hot tub, relatively uncomfortable, but, whatever you said, 45 minutes or whatever. We don't necessarily want to suggest that to people. Is that right?
Yes. I always say when I give talks, I am not an MD, I am a PhD, I am not prescribing this. We're a long ways from that. And it's definitely the case that there may be some populations that this isn't safe for. We know that heat shock proteins have been studied. They do strange things in cancer cells, for example. They actually are activated in cancer cells, so you might want to be cautious there. Neuropathy, where maybe the peripheral pain and heat receptors are not as effective, maybe there's some risk there. So definitely we need to figure out, which populations, might be less safe for these interventions, for sure.
Oh, okay. All right. I was actually talking about saying if people on a high fat diet or eating crappy food, we don't want to be saying you can do that. That's fine as long as you jump in a hot tub every, once a week.
Gotcha. Yes. No, it's similar to, you can't outrun a bad diet, right? You can't out-heat a bad diet, I would say.
There you go. All right. And you touched on age there as well. So is there any sort of interesting thing going on there with aging?
Yeah, so limited knowledge about this in terms of what's out there in the literature, but we did an aging rodent study, and we found that we could activate the heat shock proteins just as well. And we found that we could induce a glucose uptake with a one heat bout in aged rats.
Okay.
And, you know, there's some other literature out there suggesting that the heat shock protein levels are lower with age, but you can still get that activation and induction. And certainly we're doing some studies now in older individuals, and they tolerate the heat really well.
Okay.
And again, whether or not we'll see as much effect on some of the outcomes that we're looking on, you know, we don't know yet, but I'm hopeful that we will.
Even this fits with, you know, there's more and more coming out that with aging, responses to exercise are quite often normal. It's just they don't exercise as much. So, you know, I think there's more intact than we necessarily think. One thing that's been in the back of my mind is we kind of assuming, I guess, that the heat shock proteins are what are having these effects. So when you have heat, you get the heat shock proteins, and then you get the mitochondria, the insulin sensitivity, the muscle remodeling, whatever. Is there anything else going on with the heat? So you touched on blood flow at one stage.
Yeah, that's a great question. My bias is obviously that heat shock proteins are the main mediators of all these effects, but that's a very good point that there could be other things happening. In terms of blood flow, you know, again, the heat shock proteins may be involved, but it could be, you know, a nitrous oxide mediated mechanism that is independent of heat shock proteins that, you know, improves things like shear stress and vascular function. There are some studies suggesting that you're changing membrane permeability with the heat, things like lipid bilayers, lipid rafts, things like that could be changed with the heat that may have effects on things, you know, like glucose uptake for sure. So definitely think that there are other possibilities at work. Just don't know for sure.
Yeah, I guess it's like, again, everything's complicated in physiology and exercise and heat. I'm sure there's more going on than we realize. One thing I'd like to ask you about is, you know, people tend to think, you know, you do research, you get these great findings, you write a paper and off you go, but things don't always go to plan, right? Often you've got to troubleshoot, you've got to work out what's going on, or the study doesn't work out or whatever. Could you give us an example of where that's happening with you, or maybe everything's always gone to plan?
Definitely not always gone to plan. We could do a whole nother hour-long podcast if you have the time.
Yeah.
You know, I think there's been a lot of things we've done that, you know, I would have loved to have seen come, you know, to fruition. You know, there were things we wanted to do where we're isolating the skeletal muscle and heating just the leg and getting at this idea of tissue specific versus a systemic effect when we were working with our rodent model. You know, we used to take heating pads and take them apart and try to make a mini rodent heat coil, things like that, you know, to try to get at, you know, things like that we've things like that we've tried to jerry rig in the lab to get at some of those questions, and some of, you know, none of those ever worked very well. Certainly we've done some studies that we hoped would show bigger effects. We spent a lot of time working on a tissue specific knockout model of one of the heat shock proteins, and we knocked it out in the liver and didn't make a whole lot of difference. And getting back to your question earlier about redundant systems, and there's a lot of these heat shock proteins. So even though some studies, you know, Mark Fabrio's group has shown that if you overexpress heat shock protein 72 in the muscle, you get 50% increase in mitochondria, and you get these mice that can run for twice as long on a treadmill and things like that. So we thought for sure we would see an effect in the liver, and we saw very little effect on mitochondria in the liver when we knocked out that heat shock protein. So that was very disappointing. We did see some nuanced effects on glucose overall, whole body glucose regulation. So we do think it plays a role, but definitely in the liver, it didn't seem to play as critical of a role as it did in the muscle. And that was definitely disappointing.
Okay. Well, I guess you mentioned the liver a few times. So what was your thinking there? Because I know you started off, you said you were looking at muscle. I think you said your initial studies before you went to John Lossey's lab was, why don't you tell us about that, your muscle, and then how you ended up being interested in the liver, I guess.
Yeah, so I consider myself a muscle physiologist. I did a lot of single fiber work as a graduate student. I've got seduced into liver physiology from a colleague, these collaborations, how that goes.
Yeah.
John Tifo came to KU from Mizzou and came down the hall and started doing all this great work in the liver. And it just seemed like a good avenue. A lot of people weren't looking at the liver and heat shock proteins, but we know that the liver is very responsive to exercise. And so it just seemed like, and there's a lot of cool tools you can do in the liver that you can't do in muscle. You can get overexpression or ablation of a protein by just injecting in the tail vein with the liver, and you can't do that in muscle. It's a lot harder to get a tissue specific change in protein expression, for example. So there were some tools that were available to us that allowed us to do those studies, and we're still doing some of that. And I think it's interesting in heat shock proteins and heat in general, we don't know much about the way the different tissues respond. So I just think it was a natural transition. We've also done some work in the adipose tissue for the same reason.
So that's an interesting point then. So if you think about insulin sensitivity, again, I tend to be a muscle person. So I think about, exercise or heat, increasing insulin sensitivity by affecting the muscle, because the muscle is the major site of glucose uptake. But what do we know about the liver there? So, because the liver is also insulin sensitive, and, diet and exercise affect liver's insulin sensitivity. What do we know there?
Right. Right. So we know that diet and exercise impact the ability of the liver to, produce glucose and to, maintain whole body glucose homeostasis. And so, our interest shifted to fatty liver disease and how exercise and heat might impact fatty liver. And so we know that a lot of the same processes, insulin action, mitochondrial function that we think of in muscle, a lot of those same systems are in play in the liver, but again, with a slightly different purpose, right? The muscle is taking up glucose, the liver is producing glucose. So you could imagine that those same pathways are having slightly different effects depending on, the model that you're using.
Yeah. All right, so we'll start to finish up. One thing I was thinking about, I came across someone talking about their book. I can't remember their name now, but they were talking about how we're so comfortable nowadays. It's the problem of too much comfort or something. There's a book about that. And I was just wondering about with us, we're so used to being like plus or minus 1 degree, like if you put the aircon on, you put the heating on, you're just always in this perfect sort of thing. My feeling is that can't be good for you, because we know that heat has beneficial effects, but we also know, and we haven't talked about it, but cold can also have beneficial effects, with brown fat and beige fat and whatever. What do you think about that? Do you think we should be less comfortable, in terms of thermoregulation?
Yeah, I do. And I think, I like to think of heat, cold, and exercise as all stress responses that can be good for you. And, going back to my favorite, phrase that I've been using this whole time, cold stress turns on heat shock proteins too. So the reason they're misnamed, because they're stress proteins. So cold stress can turn them on as well. So yeah, I think that, we've sort of been conditioned to think of exercise as being good for us, but it's also definitely a stress and something that can be quite uncomfortable. But yeah, it's good for us. So I think, we need to shift towards thinking that way about environmental stressors, about heat. We battle with this idea as I'm writing grants on some of these applications of heat therapy. I run into this idea that too much heat is not a good thing, right? Everyone that's ever gotten into a hot tub at a hotel sees those warning signs. Don't stay in longer than 15 minutes, right? We are conditioned to think that that is harmful. And really, it's just uncomfortable.
Yeah, so obviously this is probably playing a role in why everything's so messed up, why we've got so much obesity and so much heart disease and so much cancer and everything, because we're not meant to be comfortable the whole time. We're meant to be hunter-gathering, so actually being active, trying to get food, and then, shivering in the cave, and then other times we're hot and we're just too comfortable. Yeah, I think that book, maybe I should have a look at that book, whatever it is. All right.
Yeah, these are physiological responses. Absolutely.
All right. So is there anything, I think we've covered a fair bit. Is there anything you'd like to get out before we finish up? It's been great.
Yeah, no, when are you going to get your hot tub?
Yeah, exactly. I've got a bath, but what is the difference? Hang on, a bath and a hot tub. The hot tub is when you can turn on the bubbles, I guess.
Yeah, and the hot tub usually has some kind of you know, motor that's going to keep the heat up.
Oh, like
You know,
circulating.
yeah, and keep
Ah,
that steady temperature versus a bath, once you've run the water, it's going to start cooling down. So
and
that's
get
the
that
big
boundary
difference.
layer. You also get that boundary layer. You got to keep moving it. So when you hurt your ankle and you put it in ice, if you just leave it there, it doesn't feel that cold. You've got to keep it moving around. Yeah. Okay.
Yeah.
Okay. Maybe I need to get a help. I'll talk to my wife about it.
There you go.
Sounds good. Okay. Well, thanks again for coming on. Actually, are you okay if people contact you? Are you on Twitter or?
Yeah, sure.
All right. So how would they
Yeah.
contact you?
On Twitter, I'm at Paige Geiger, PhD. Yeah, so
All right. Cool.
there's a lot of good science on Twitter these days. So I try to keep up a little bit.
And there's also a lot of crap on there as well, actually. That's part of the plan with this podcast is instead of people getting their information from influencers, why not get it from the experts such as yourself? Yeah.
Excellent.
Okay.
Yeah,
Right.
well, thanks for giving me the chance to talk about this. This is fun.
Thank you. I've learned a lot, so it's been great. Okay. See ya.
All right, bye.
Bye.